Cell cycle and cell division. In this chapter, we will read about cell cycle, M phase, significance of mitosis, meiosis, significance of meiosis. Are you aware that all organisms, even the largest, start their life form from a single cell? You may wonder how a single cell then goes on to form a such large organism. Growth and reproduction are characteristics of cells, indeed of all living organisms. All cells reproduce by dividing into two with each parental cell giving rise to two daughter cells each time with they divide. These newly formed daughter cells can themselves grow and divide, giving rise to a new cell population that is formed by the growth and division of a single parental cell and its progeny. In other words, such cycles of growth and division allow a single cell to form a structure consisting of millions of cells. Cell cycle. Cell division is very important process in all living organisms. During the cell division of a cell, DNA replication and cell growth takes place. All these processes, that is cell division, DNA replication and the cell growth, hence have to take place in a coordinated way to ensure the correct division and formation of a progeny cells containing intact genomes. The sequence of events by which a cell duplicates its genome, synthesizes the other constituents of the cell and eventually divides into two daughter cells is termed as cell cycle. Although cell growth, that is in terms of cytoplasmic increase, is a continuous process, DNA synthesis occurs only during one specific stage in the cell cycle. The replicated chromosome, that is DNA, are then contributed to the daughter nuclei by a complex series of events during a cell division, and these events are themselves under genetic control. Phases of cell cycle. A typical eukaryotic cell cycle is illustrated by human cells in culture. These cells divide once in approximately every 24 hours. However, this duration of cell cycle can vary from organism to organism and also from cell type to cell type. Yeast, for example, can progress through the cell cycle in only about 90 minutes. The cell cycle is divided into two phases. First, interphase and second, M phase. The M phase represents the phase when the actual cell division or mitosis occur, and the interphase represents the phase between the two successive M phases. It is significant to note that in 24-hour average duration of cell cycle of a human cell, cell division proper lasts for only about an hour. The interphase lasts for more than 95% of the duration of the cell cycle. The M phase starts with the nuclear division corresponding to the separation of daughter chromosome, that is karyokinesis, and usually ends with this division of cytoplasm, that is cytokinesis. The interphase, thou called the resting phase, and is the time during which the cell is preparing for division by the undergoing both the cell growth and DNA replication in an orderly manner. The interphase is divided into three further phases. First of all, the G1 phase, that is gap 1 phase, and second S phase, that is synthesis phase, and the third G2 phase, that is gap 2 phase. G1 phase corresponds to the interval between the mitosis and the initiation of DNA replication. During the G1 phase, the cell is metabolically active and continuous grows but does not replicate its DNA. S or synthesis phase marks the period during which the DNA synthesis or replication takes place. During this time, amount of DNA per cell doubles. If the initial amount of DNA is denoted as 2C, then it increases to 4C. However, there is no increase in the chromosome number if the cell had diploid or 2n number of chromosome at the G1. Even after S phase, the chromosome number remains the same, that is 2n. In the animal cells, during the S phase, DNA replication begins in the nucleus and the centriole duplicates in the cytoplasm. During the G2 phase, proteins are synthesized in the preparation for the mitosis while cell growth continues. Some cells in the adult animals do not appear to exhibit division, example heart cells, and many other cells divide only occasionally as needed to replace the cells that have been lost because of injury or the cell death. And these cells that do not divide further exit G1 phase to enter an inactive stage called the quiescent stage, that is G0 of the cell cycle. Cells in this stage remain metabolically active but no longer proliferate unless called on to do so depending on the requirement of the organism. In animals, mitotic cell division is only seen in the diploid somatic cells. However, there are few exceptions to this where haploid cells divide by mitosis. For example, male honeybees. Against this, the plants can show mitotic division in both haploid and diploid cells. From your recollection of example of alternation of generation in plants, identify plant species at which mitosis is seen in haploid cells. M phase. This is the most dramatic period of the cell cycle, involving a major reorganization of virtually all components of the cell. Since the number of chromosomes in the parent and progeny cells is the same, and it is also called as equational division. Thou, for the convenience, mitosis has been divided into 4 stages of nuclear division, that is karyokinesis. It is very essential to understand that cell division is a progressive process and very clear-cut lines cannot be drawn between various stages. Karyokinesis involves the following 4 stages, that is prophase, metaphase, anaphase, telophase. First of all, we will see the prophase. Prophase, which is the first stage of karyokinesis of mitosis, follows the S and the G2 phases of the interphase. In the S and the G2 phases, the new DNA molecules formed are not distinct but intertwined. Prophase is marked by the initiation of condensation of chromosomal material. The chromosomal material becomes untangled during the process of chromatin condensation. The centrosome, which had undergone duplication during S phase of interphase, now begins to move towards the opposite poles of the cell. The completion of the prophase can be marked by the following characteristic events. Chromosomal material condenses to form compact mitotic chromosomes. Chromosomes are seen to be composed of two chromatids attached together at the centromere. Centrosome, which had undergone duplication during the interphase, begins to move towards the opposite poles of the cells. Each centrosome radiates out microtubules called the asters, and the two asters together with the spindle fibers forms mitotic apparatus. Cells at the end of prophase, when viewed under the microscope, do not show Golgi complex endoplasmic reticulum, nucleolus, and the nuclear envelope. Metaphase. The complete disintegration of nuclear envelope marks the start of the second phase of mitosis. Hence, the chromosomes are spread through the cytoplasm of cell. By this stage, condensation of chromosome is completed and they can be observed clearly under the microscope. This then is the state at which the morphology of chromosome is most easily studied. At this stage, metaphase chromosomes is made up of two sister chromatids which are held together by the centromere. Small disc-shaped structure at the surface of the centromere are called kinetochores. These structures serve as the site of attachment of spindle fibers that is formed by the spindle fibers to the chromosomes that are moved into position at the center of the cell. Hence, the metaphase is characterized by all the chromosomes coming to lie at the equator with one chromatid of each chromosome connected by its kinetochore to the spindle fibers from one pole and its sister chromatid connected by its kinetochore to spindle fibers from the opposite pole. The plane of alignment of the chromosome at the metaphase is referred to as metaphasic plate, and the key features of the metaphase are as spindle fiber attached to the kinetochores of chromosomes. Chromosomes are moved to the spindle equator and get aligned along the metaphase plate through the spindle fibers to the both. Anaphase. At the onset of anaphase, each chromosome arranged at the metaphase plate is split simultaneously, and the two daughter chromatids, now referred to as the daughter chromosomes of the future daughter nuclei, begin to air migration towards the opposite poles. As each chromosome moves away from the equatorial plate, the centromeres of each chromosome remains directed towards the pole and hence at the leading edge, with the arms of the chromosome trailing behind. Thus, anaphase is characterized by the following key events. Centromeres split and chromatids separate. Chromatids move to the opposite poles. Telophase. At the beginning of the final stage of karyokinesis, that is telophase, the chromosomes that have reached their respective poles decondense and lose their indivisibility. Their individual chromosomes can no longer be seen, and each set of chromatin materials tend to collect at each of the two poles. This is the stage which shows the following key events. Chromosomes cluster at the opposite spindle poles and their identity is lost as discrete unit elements. Nuclear envelope develops around the chromosomes cluster at each pole, forming the two daughter nuclei. Nucleolus, Golgi complex, and ER reform. Cytokinesis. Mitosis accomplishes not only the segregation of duplicated chromosome into daughter nuclei, that is karyokinesis, but the cell itself is divided into two daughter cells by the separation of cytoplasm called cytokinesis, at the end of which division gets completed. In an animal cell, this is achieved by the appearance of a furrow in the plasma membrane. The furrow gradually deepens and ultimately joins in the center, dividing the cell cytoplasm into two. Plant cells, however, are enclosed by a relatively inextensible cell wall. Therefore, they undergo cytokinesis by a different mechanism. In the plant cells, wall formation starts in the center of the cell and grows outward to meet the existing lateral walls. The formation of new cell wall begins with the formation of a simple precursors called the cell plate that represent the middle lamella between the walls of the two adjacent cells. At the time of cytoplasmic division, organelles like mitochondria and plastids get distributed between the two daughter cells. In some organisms, karyokinesis is not followed by the cytokinesis, as a result of which multinucleate condition arises, leading to the formation of syncytium, example liquid endosperm in the coconut. Significance of mitosis. Mitosis or the equational division is usually restricted to the diploid cells only. However, in some lower plants and in some social insects, haploid cells also divide by mitosis. It is very essential to understand the significance of this division in the life of an organism. Are you aware of some example where you have studied about haploid and diploid insects? Mitosis usually results in the production of diploid daughter cells with identical genetic complement. The growth of multicellular organism is due to mitosis. Cell growth results in the disturbing the ratio between the nucleus and the cytoplasm. It is therefore becomes essential for a cell to divide to restore the nucleocytoplasmic ratio. A very significant contribution of mitosis is the cell repair. The cells of the upper layer of the epidermis, cells of the lining of the gut, and the blood cells are being constantly replaced. Mitotic division is the meristematic tissue. The apical and the lateral cambium result in the continuous growth of the plant throughout their life. Meiosis. The production of offspring by the sexual reproduction includes the fusion of two gametes, each with a complete haploid set of chromosomes. Gametes are reformed from specialized diploid cells, and this specialized kind of cell division that reduces the chromosome number by the half result in the production of haploid daughter cells. This kind of division is called meiosis. Meiosis ensures the production of haploid phase in the life cycle of sexually reproducing organisms, whereas fertilization restores the diploid phase. We come across meiosis during gametogenesis in plants and animals. This leads to the formation of haploid gametes, and the key feature of the meiosis are as follows. Meiosis involves two sequential cycles of nuclear and cell division called the meiosis 1 and the meiosis 2, but only a single cycle of DNA replication. Meiosis 1 is initiated after the parental chromosome have replicated to produce identical sister chromatid at the S phase. Meiosis involves pairing of homologous chromosomes and the recombination between non-sister chromatids of the homologous chromosomes. 4 haploid cells are formed at the end of meiosis 2. Meiotic events can be grouped under the following phases. Meiosis 1, in meiosis 1, prophase 1, metaphase 1, anaphase 1, telophase 1. In meiosis 2, prophase 2, metaphase 2, anaphase 2, telophase 2. Now we will read about the meiosis 1. In the meiosis 1, first of all, we will see prophase 1. Prophase of the first meiotic division is typically longer and more complex when compared to prophase of mitosis. It has been further subdivided into following 5 phases based on the chromosomal behavior, that is leptotin, zygotin, pachytin, diplotin, and dikinesis. During the leptotin stage, the chromosomes becomes gradually visible under the light microscope. The compaction of chromosomes continue throughout the leptotin. This is followed by the second stage of prophase 1 called the zygotin. During this stage, chromosomes start pairing together, and this process of association is called synapsis. Such paired chromosomes are called homologous chromosomes. Electron micrographs of this stage indicate that chromosome synapsis is accompanied by the formation of complex structure called synaptonemal complex. The complex formed by a pair of synapsed homologous chromosomes is called a bivalent or a tetrad. However, there are more clearly visible at the next stage. The first 2 stages of prophase 1 are relatively short-lived compared to the next stage, that is pachytin. During this stage, the 4 chromatid of each bivalent chromosomes become distinct and clearly appears as tetrads. This stage is characterized by the appearance of recombination nodules, the site at which the crossing over occurs between the non-sister chromatids of the homologous chromosomes. Crossing over is the exchange of genetic material between the homologous chromosomes. Crossing over is also an enzyme-mediated process, and the enzyme involved in is called the recombinase. Crossing over leads to a recombination of genetic material on the 2 chromosomes. Recombination between the homologous chromosomes is completed by the end of pachyting, leaving the chromosomes linked at the site of crossing over. The beginning of the diplotin is recognized by the dissolution of the synaptonemal complex and the tendency of a recombined homologous chromosomes of the bivalents to separate from each other except at the site of crossing overs, and the except structures are called the chiasmata. In the oocyte of the some vertebrates, diplotin can last for the months or the years. The final stage of meiotic prophase 1 is dikinesis. This is marked by the terminalization of chiasmata. During this phase, the chromosomes are fully condensed and the meiotic spindle is assembled to prepare the homologous chromosomes for the separation. By the end of dikinesis, the nucleolus disappears and the nuclear envelope also breaks down. Dikinesis represents the transition to metaphase. Metaphase 1, the bivalent chromosomes aligned on the equatorial plate and the microtubules from the opposite poles of the spindle attached to the kinetochores of the homologous chromosomes. Anaphase 1. The homologous chromosomes separate while sister chromatids remain associated at their centromeres. Telophase 1. The nuclear membrane and the nucleolus reappear. Cytokinesis follows and is called as dyad of the cells. Although in the many cases the chromosomes do not undergo some dispersion, they do not reach the extremely extended state of the interphase nucleus. The stage between the 2 meiotic division is called interkinesis and is generally short-lived. There is no replication of DNA during interkinesis. Interkinesis is followed by the prophase 2, a much simpler prophase than the prophase 1. Meiosis 2, prophase 2. Meiosis 2 is initiated immediately after cytokinesis, usually before the chromosome have fully elongated. In contrast to the meiosis 1, meiosis 2 resembles a normal mitosis. The nuclear membrane disappears by the end of prophase 2. The chromosomes again become compact. Metaphase 2. At this stage, the chromosomes align at the equator and the microtubules from the opposite poles of the spindle get attached to the kinetochores of the sister chromatids. Anaphase 2. It begins with the simultaneous splitting of the centromeres of each chromosomes, which was holding the sister chromatids together, allowing them to move toward the opposite poles of the cell by shortening the microtubules attached to the kinetochores. Telophase 2. Meiosis ends with the telophase 2, in which the two groups of chromosomes once again get enclosed by the nuclear envelope. Cytokinesis follows, resulting in the formation of tetrad of the cell, that is 4 haploid daughter cells. Significance of meiosis. Meiosis is the mechanism by which the conservation of a specific chromosome number of each species is achieved across generation in sexually reproducing organisms, even though the process per se paradoxically result in the reduction of chromosome number by half. It also increases the genetic variability in the population of organisms from one generation to the next. Variation are very important for the process of evolution.